Hello and welcome to this next session where I'm honored to have with me Dr. George Church for a fireside chat about resilience to all viruses. I'm Rion Prior, a former writer for CNN and the author of the upcoming book, The Long Haul, which looks at long COVID and other long-term effects of the COVID-19 pandemic. I'm excited to dive into a similar topic with George related to this ongoing conflict between viruses and humanity and how we can guard ourselves against future threats. Dr. Church is a professor of genetics at Harvard Medical School. He's a founding member of the Wyss Institute and the director of personalgenomes.org, the world's only open access information on human genomic, environmental, and trait data. He is known for pioneering fields of personal genomics and synthetic biology and developed the methods for the first genome sequence and dramatic cost reductions since then. He's contributed to nearly all next generation sequencing methods and companies. His team was a part of inventing CRISPR for human stem cell genome editing and other synthetic biology technologies. And he's co-authored more than 590 papers and has 155 patent publications, including as well as one book, Regenesis. His honors include the Franklin Bauer Laureate for Achievement in Science, the Time 100, and election to the National Academies of Science and Engineering. Dr. Church, thank you so much for joining me.
Oh, thank you. It's great to be here, Ryan.
And we'll start off with a broad question about a topic that's been on most people's minds for the past 2 years. From your point of view, what are the biggest challenges in the past 2 years for overcoming the COVID-19 pandemic? And what are the most important lessons learned that you would like to bring to the attention of this audience?
Well, I had been advocating since well before COVID that we should be prepared. We should be constantly testing because the cost of testing is incredibly low, looking for new emerging viruses. And we should be ready to have masks, because that's a very cost-effective and general way. It works no matter what the strain is. And we should always be prepared. We shouldn't have supply side line issues. So I think these are things that we have learned. There are vaccines have also been quite impactful, and I think we learned, in addition to the obvious lessons about how quickly we can bring out a new vaccine, even a new category of vaccines like the messenger RNA vaccines, or variations on gene therapy tools being applied to vaccines. That got passed in 1 year, which is amazingly fast, and the lowest cost ever for gene therapies. So gene therapy is typically used for rare diseases, but when it's used for something common like a pandemic, it dropped down to $2 a dose from $2 million a dose. So these are amazing lessons that we've learned, and I hope we take them to heart going forward. They're just getting cheaper and more effective, so we really should be almost no barrier other than education.
Now, when talking about this topic, the idea of viral or disease resilience, what are the most important things to be aware of and to correctly understand what resilience means in this context, and what does that depend on, and how can we improve on that?
Well, resilience is used in many ways, but I think in this context, the interesting idea, and I'm guessing where you're coming from, is that there are some individuals who are naturally resistant to viruses and other individuals who are not, and certain species that are resistant to certain pathogens and others that are not. And so we've been, trying to exploit these and study and figure out. So for example, there's some people that are resistant to HIV because of genetics in one of their genes, CCR5. And there are people that are resistant to norovirus because of genetics in one of their genes, FUT2. In fact, half members of my family are resistant, half are not. It's about every 2 years, half of us get sick with norovirus. So this is examples of resilience that we'd like to. Also, there's animals that never get human diseases and vice versa. There are some that cross over, so-called zoonotic diseases, and some that cross over so rarely they effectively are human specific. So anyway, we want to exploit these differences and maybe make them more readily available and inexpensive.
And I'm very interested in this idea of the bio weather map and the ideas about how can regular people or citizen scientists be involved in the detecting or surveillance of viral threats, and why have you been passionate about that, and what might that mean for how we guard against future viruses?
Yeah, I've been quite passionate about this since around the turn of the millennium, and coined the term bioweather map, because I think weather map is one of the small number of citizen science things that really everybody feels that they can be engaged in. So I mean, there's things like gardening and breeding of pets and astronomy and so forth, which a few people are involved in. But weather is something that really impacts everybody. It can be life and death. If a tornado is coming your way or you slip and fall on the ice, it really is impactful. And I think to some extent, pathogens are also just as impactful and just as easy to understand. Now, it is true that they're mostly invisible in every day. You can't see them the way you see a hurricane, but to some extent, the way we relate to weather is pretty far in advance when we can't see it either. We see it through satellite weather imaging, and we need a similar way, some kind of goggles that we can see all the pathogens around us, or in our food or water, air, locally and globally, emerging zoonotic diseases that are coming out of the farm or the forest. All these things need to be displayed in some kind of entertaining, colorful map, and other interviews, and all the things that go into a good weather station should go into a good bioweather station, and engage citizen participation.
And what progress have you made in making this a reality? And what do you hope can be accomplished in the next couple of years?
Yeah, I mean, I can't, even my team and I can't take too much credit. I mean, a lot of the credit goes to the very unfortunate disease that we just had. People are getting diagnosed a lot. It's so routine. My entire university has had access to free testing. Some of us get tested daily. That some of us have actually worked on diagnostics and therapeutic COVID components during the outbreak, early in the outbreak. So that's one thing that we can build on that wasn't present when we first started this years ago. Another thing that has happened that they are responsible for is the Personal Genome Project, personalgenomes.org, where we recruit volunteers who are willing to share their story, share their genome, their medical records, their interaction with pathogens and good bacteria, their microbiome, on the internet. And so that's taken off now in multiple continents, with ethics approval in each of these countries. And so that's great, very gratifying to watch that movement grow internationally.
Yeah, and I do like this idea about resilience toward all viruses, or just resistance to viruses. How do we better improve that in like a sustainable way, in a global way, in building systems that can work as future viruses emerge?
Yeah, that's a great question. Very close to my heart as well. The sustainable and accessible, independent of wealth, is a very important component of this. There are very few technologies, whether they're conventional physics and chemistry technologies or biotechnologies, that are really equitably shared around the world. But one of them is smallpox, the extinction of smallpox in particular. So we don't need to worry about it evolving to require new drugs and new vaccines, because it's completely extinct. And so that means that every person on the planet gets to benefit from this without them or their nation shelling out any money because it's gone. And there are many pathogens that are essentially human specific, very close to it. So if we can eliminate them once, then they're gone forever. And these include things like polio and guinea worm, which are very close to extinct, very close to following smallpox in this. So that's one way of getting equitable and broad distribution of a technology. It may cost a little bit of money to get there, but far less than dealing with intensive care units and building up containment and quarantine and PPE protective equipment for hospital. Extinction of the pathogen is a good route.
How do you feel about applying this idea to COVID that where once we thought that COVID might be something that we could completely eliminate or at least battle down to a dull roar, in the next 1 to 5 years, how is our relationship with COVID going to evolve?
Well, the conventional wisdom among people who study infectious diseases is that there will be some coevolution between the host and the pathogen. The pathogen will get weak enough that it will spread and not immediately kill the host, and the host will get a little bit stronger, and it won't completely wipe out the human race. That's the good news. The bad news is there's lots more where that came from. I mean, we're seeing monkeypox now. We'll see recombinants, between viruses where two viruses that adapted to us will recombine, or one virus that's in an animal and one that's in a human will recombine and make something that's unpredictable. So I think we need to get good at, and part of the reason that it's happening more recently is we have a more well-stirred pot. There was a lot more travel than there ever was. There's a lot more invasion into natural wild environments, and there's more crowded farming going on. And those three things, the travel, the wild, humans invading the wild, and humans building up very high concentrations in cities and in farms, means that we're going to see more and more of this. And we have to be very good at it. And we should be able to just pull out masks for the whole family on a moment's notice, rather than having to spend a year building up supply chains, for example.
Right. Yeah, and it's slightly separate topic that you're a pioneer in genome engineering and in systems and synthetic biology. Can you share with us some of the latest breakthroughs with your research and work, and what are you most excited about, and what major challenges do you feel are ahead?
Oh, yeah. So I'll start with one that's very relevant to this, but I have to preface that with most of the stuff that we work on sounds like science fiction when we start working on it, and it sounds like is fully accepted routine stuff by the time the public hears about it. So, for example, we helped develop two major ways of doing sequencing, of reading out, of doing diagnostics. One of them is fluorescent microscopy, and the other one is nanopore sequencing. And both of those sounded like crazy when we first started working on them in the 80s. And
now they're applied.
Now they're routine. They work really well. Nanopore is particularly good at reading long distances, and the fluorescent microscopy is so general that it actually can work for non-invasive prenatal testing. It's used for looking for rare diseases. It's used for finding emerging diseases, and you can even use it for microscopy, which is where it came from. A whole new category.
Finding microclots in long COVID has been one.
Yep. So it's a very versatile tool. It's even used for things outside of biology. It's used for forensics. It's used for data storage. So anyway, that was just the preface of saying, I'm going to say something that's going to sound like science fiction, but it's very close to reduction to fully reduced to practice. And that is that we have a way that we've been working on, I don't know, since at least 2009, to make any species resistant to all viruses, all natural viruses, even ones we've never seen before. And that is based on the genetic code, the code that goes from DNA to RNA to proteins. And that code consists of all possible triplets of the A, C, G, and T that are the 4 bases. You can combine any 3 of them in 64 ways. So, for example, AAA codes for an amino acid lysine. So that's when people talk about the code, that's what we're talking about. And so we've learned how to change the code. We did that as long ago as 2013. We changed the code and found that made, as we expected, it made the organism being genome engineered resistant to almost all viruses. Not quite, maybe 90% of them. And then since 2013, we've been working on new improved versions of this that makes that same organism completely resistant to viruses. And to prove that, we take viruses from the wild, random viruses we know nothing about, and challenge. We've taken some from the local river and from the local farm, and we now think we have one organism that is completely resistant to all natural viruses, both for theoretical reasons and by these tests that I mentioned. And now we're moving on and trying to apply this to other organisms, because we now know how to do it quickly, and we're trying to apply it to plants, agricultural species, plants, animals, and then human cells, tissues, organs.
No, and it caused me to do a double take that, that it does sound like science fiction. And
but we delivered on 90% of it in 2013, and we'll be publishing a paper later this year that shows full virus resistance.
For one organism.
For one organism. So it's an industrial microorganism, particularly important and easy one, but we're going to use exactly the same tools in a variety of organisms that we care about.
And we'll just what makes you excited about this?
Well, it's 2 reasons. I mean, one is that there's more to it than that. I mean, it not only gives us this virus resistance, it also gives us a whole new set of chemistry and new amino acids, which you don't need for the virus resistance, but it comes along as a bonus, and a biocontainment method, which could be important in certain applications where you want to release things into the wild. This would a typical application, you would not be releasing into the wild, but we have that option. So we get 3 things for the price of 1, which is the recoding of the genome. That's one reason exciting. Also, just philosophically, it's an interesting idea that almost all of our anti-pathogens, antivirals, have been very hard won. It'll be say an anti-HIV drug that's antiviral, and before you know it, the virus is resistant to it. So then you have to give it like bunch of new drugs and do them all at once, the multi antiretroviral therapy, or ART, and it's hard. Or we'll find one gene at a time, like I was talking about at the beginning, where humans naturally occurring in our population will have, by luck or possibly by selection, become resistant to HIV or norovirus or other ones. And then we look in there for clues for how we could transfer that superpower to other people. But each of these is one virus at a time, sometimes one strain of virus at a time. So a vaccine might work for one strain, but then it evolves away from it, as we've sort of seen with COVID-19. So each of these has been hard won. So here, what seems so exciting and surprising is that we can come up with a method that should work for all organisms and all viruses. I mean, you still have to do it one organism at a time, but for each of those organisms, it's the same plan, and we get better and better at it. And once you do it, it works for all viruses. Now, that's not all pathogens. I should note, it's all viruses, including pathogenic viruses.
And I wonder, well, this is a huge question too, but what are the risks of doing this with where obviously there could be situations where a virus could be good, and then protecting against all viruses that you might put that back.
Yeah. So you need to test all things. All new drugs, all new medical devices have to go through the FDA for testing, and this will be no exception. If you're using it even in a farm animal, it would not only have to get FDA approval, but also USDA approval, and maybe even EPA, meaning agricultural and environmental weigh-in. This is all good. Those are great organizations, and I think they deserve our kudos for the job that they do. And this will go through those steps. I think the scenario that I've my team and I have tried to address in advance. So a lot of these new technologies we've developed that sound science fiction-y, it's nice to have them in the science fiction phase where we can work with screenwriters for movies to portray a negative scenario, to in a certain sense inoculate ourselves from that negative scenario by talking about it, by imagining all the ways it can go wrong, all the Andromeda Strain, Jurassic Park, Frankenstein scenarios, and not so we'll be so that we'll ignore it when it comes, but so we'll be ready for it when it comes, ready for it well before it comes, all these negatives. So in this case, I think that if you made a microorganism that were resistant to all of its viruses, then it would have a selective advantage over other microorganisms. And so you want to make sure that microorganism is biocontained. So that's why it's good that this recoding gives you all three of these things, and one of them is biocontainment. You can make it so that it can only grow if you give it a chemically manufactured food component. This is actually also out of Jurassic Park, where they used lysine, which was not a good choice because that's present in all foods. But we have another amino acid, not lysine, called BIP, which is present in no foods. It's only made by organic chemistry. So, that's an example of the cool new solution, the possible problem with the cool new solution, the possible solution to that problem. And you keep talking about it and engaging more people to make sure that we've thought through these things, and then test them very carefully in physically and biologically contained test beds. Now, if we're making human tissues, that's not an issue. Human tissues are not going to escape into the environment and have a selective advantage, but they could be extremely valuable medically.
No immediate risk of these becoming an invasive species.
Well, we already had, the bad news is humans already are an invasive species. 96% of mammals, this is a cool fact you can tell your friends, 96% of mammals on the whole planet, not just North America, are now humans in their agricultural species and pets. 96%. And 77% of crops are used for feeding them, not for feeding humans. So we're an invasive species if ever there were one. But we like us. We like us.
Weird. I wanted a tempting question, mentioning Jurassic Park, and I know a passion of yours has been the idea of resurrecting extinct species. And where are we or where is your team or researchers in the process of where a woolly mammoth might be possible to walk among us?
This probably takes a little more time than we have, but I'll give you the short version, which is we're not really trying to bring back new species so much as create diversity by using information from ancient DNA. So we have brought back ancient genes and shown that they have properties that are consistent, say, with cold resistance. And there are a few keystone species that using combination of this diversification, we can save an endangered species or help rewild a keystone species. The most famous example of that is it's probably the wolves at Yellowstone, which were gone for 70 years. And when there were various reasons, theoretical reasons to restore them. We tried restoring them and it worked great because it set off a very positive chain reaction that ecologists, I think, like to use as a textbook now, is that the wolves changed the large herbivore population, which changed the willows and other trees, which changed the beavers, changed the lakes and the fish. And it all had this very positive balance now that was missing when the wolves were gone. The same thing's true for Arctic elephants. There used to be elephants on almost every continent of the world, and we wiped them out except for two endangered species, two categories of endangered species in Africa, Asia, and it's particularly potent, not just from an ecological standpoint, but from a very human, selfish standpoint, is that the Arctic is melting, and when it melts, as it melts, it's releasing up to 1,400 gigatons of carbon dioxide and methane being 30 to 80 times worse than carbon dioxide, and 1,400 gigatons being much worse than the 10 gigatons of all human activity put together per year. So it just dwarfs the 1,400 gigatons dwarfs the 10 gigatons that if all humans stopped all activity, left the planet, we would only save 10 gigatons a year. So anyway, it's a long story about how it is that elephants help knock down trees, and that restores the grasses that resulted in a much richer ecosystem, much better trapping of carbon out of the air, better transfer of cold wind in the winter, minus 40 degrees, down to keep the huge, thick, carbon-rich topsoil, more carbon stored in the Arctic than the rest of the forests of the world put together. So anyway, you asked the progress. The progress is very simply, we went from almost no funding for this project for the last decade to very adequate funding, from about $10,000 a year to $75 million almost overnight in the last few months. So we're very appreciative to everybody that helped make that happen, and the science is going much faster than ever.
And the idea is that this isn't just a fun pipe dream. This has huge ramifications for how we deal with climate change.
I think there's a lot of enthusiasm for it, both in terms of carbon credits, in terms of new technology that could be applicable to other veterinary and endangered species. There's enthusiasm for the biodiversity that can come about when we're instead of carving up herds so that they become inbred by putting in pipelines and roads, we're increasing diversity by bringing alleles from all over the world and from back in time, as far back as a million years is the furthest back we've gone so far, getting ancient DNA.
I know we're going a little bit over time, but I want to, if I could, I'll just ask you one last question.
Sure.
As I just mentioned earlier, before speaking about this book, The Code Breaker by Walter Isaacson, which does a great job of profiling Juniper Doudna and the development of CRISPR technology, which leads to or co-winning the Nobel Prize, and your character in the book. And if people are interested in hearing more, I would recommend them reading that book to learn more about you and about synthetic biology. But I wanted to just ask, what was your experience like being interviewed for and working with Walter Isaacson? And yeah.
Walter, I had already read some of his books before, and he's an exceptional combination of a top academic scholar who really does his research, has all the footnotes and everything, but also engaging for a broad audience. I read that book in audible form as well as in the written form, and it's a real page turner. I think independent of how close you are to the events. So I think he did a spectacular research job, as he has on all of his biographies. He's a Tulane professor, and he really gives professors a good name.
Cool. I guess maybe we'll end it there. If, unless you have any other last notes to add while we're chatting.
Yeah, there's hundreds of hours more to go.
Well, I would
stay
love
tuned to this channel.
I would love to. Yeah, as I'm working on more journalistic projects, I would love to be in touch. And
okay, yeah. You bet.